Thank you very much. I'm showing you the PhD work that I did at UCL in Alison Zoid's lab, and working together with Jemima, who's here, and Ian, where in the EM facility at the LNCB, we do SEM array tomography. And the two things that I'm showing you today, where I used this technique, was to look at quite different things, but still in the peripheral nervous system. So the first one on the left is looking at the vasculature of the blood-nerve barrier, similar to the blood-brain barrier in the central nervous system that protects the peripheral nervous system. And it's very important, this microenvironment of these vessels that induce these barrier properties. So we better wanted to understand the cells, and specifically the endothelium and the pericyte, of how they interact with very pleomorphic structures, non-repetitive, penetrating the basement membrane or not. So quite a difficult challenge to segment later on. And then at the same time, we were also interested more broadly in the main glial cells of the peripheral nervous system, Schwann cells, and specifically the ones that create myelin. So they have very clear boundaries, clear structures, and uniform repetitive structures. So the first thing that I did once we acquired these data sets was to ask and see how do we best segment that. And I think this publication was really useful, summary of what is out there, and that there is still no one-size-fits-all solution, and that there's different program software. So I started with MIB, which was very useful, and then luckily we got the WebNosos toolkit in the lab and in the building. So we started using WebNosos as well as Amira back in the day. So for the first challenge, where we had to do the segmentation of the endothelium and pericytes, which was, as I described before, interdigitating cells, sometimes hard for a human eye to discern what is pericyte, what is endothelium, where the 3D nature of it really helped, where you could go through the data and see, okay, actually this little protrusion is a pericyte, so let's go and fix that. And there it was mainly manual segmentation, with the hope to then later on use all this data for as a ground truth to train a model to be able to segment more blood vessels. And then at the same time, when we were looking at Schwann cells, WebNosos has this really useful AI-powered tool, Quick Select, that we just saw before, where you can just literally click and it segments 16 slices, really easy, very minimal proofreading or correcting, because it works really well for our Schwann cells. So yeah, in summary here, one of my challenges was very labor intensive, and the other one still was much faster, but still not automated. And then what I also wanted to show you is that the next step was then I wanted to quantify these things, and these softwares have to some extent inbuilt readouts such as volume and surface area, which is helpful to some extent. But as soon as it goes a bit more complicated, or I want to, for example, understand the coverage, that wasn't inbuilt. So I had to export my data. I created 3D TIFF files, and I mostly use MATLAB. So that meant I needed to know to some extent how to code, which I didn't know. So quite fun, but still not very friendly for people who don't know how to code. So in summary, I didn't include anything about alignment here, but I think this is something important as well that we've heard before, and that WebNosos to some extent can do now. But the main challenges I've found, and I wanted to share with you, and I'm happy to discuss it later as well, or if you have any input, is that once I arrived at the segmentation point, was that what do I even use, or how can I better automate it? Then at the transition from once I segmented my data to quantifying it, is that on WebNosos I had gigabytes of data. Do I export it? Do I have to downsample it? Or is there a way that I could keep it in a cloud somewhere and access it to then quantify it? And then finally, once it goes into quantification, it goes quite quickly into knowing how to code, because of the nature of wanting to quantify things more in a customized manner. So with this, I would like to thank all of you, and I'm happy to chat later. Obviously, Alison, Lloyd's lab, our EM facility, all the collaborators and people that know how to code and to always help me. And yeah, thanks.